Thanks to Jan and the members of the organizing committee for invitation. Hello, everyone. My talk title is The Early Cerebellar Circuit Formation. Trigeminal derived pioneer axon targets the cerebellar nuclei. I will go through the early cerebellar development, overview on the cerebellar primordium, neurogenesis, axonogenesis, and relation to the trigeminal system. And then I will show you some data regarding trigeminal nerve-derived cerebellar afferent and early cerebellar afferent target neurons. Well, our cerebellum, with this complicated connectivity in adult, they develop from the early stage of the embryonic development in neural tube, the area we call rhombomerevon, alar plate of the rhombomerevon. And we know, and we have a huge number of the information regarding the connectivity in the adult cerebellum. However, the developmental mechanism that generate this complex cerebellar connectivity is not understood yet. And my focus today will be on this part. And to understand what is the relation between the trigeminal system and ganglion to the cerebellar primordium and development, and what is the target, what is the earliest afferent, and then target neuron in the cerebellar primordium. Well, the earliest connectivity is important because they provide the foundation for our neural network that our brain function depend on, and also they play as a scaffold for the incoming axon to the developing cerebellum or other part of the nervous system. So to understand the relation between the trigeminal system and the cerebellar primordium, and what is the earliest afferent to the cerebellar primordium, or what is the target neuron in here, we need to go through this early stage of the neural tube development. As we can see, I'm going to take a laser point in here. As we can see in here, this is the rostral end, and this one is the prosencephalon. And here is we have a mesencephalon. This is the rhombencephalon, and continue caudally to the spinal cord. Our rhombencephalon has, in fact, a transverse subdivision or segmentation called rhombomer from the rhombomer 1 to rhombomer 7 or 8. And here is the rhombomer 1, and rostral to this one, we have a mesencephalon, and the boundary between this is the isthmus. And this one play as one of the organizer at this stage, we call isthmic organizer. And in addition of this division, our neural tube divide ventrally and dorsally by this red dashed line that we call this one as a sulcus limitans that divide to alar plate dorsally and basal plate. Alar plate producing all neuron in sensory pathway, and basal plate, they in fact produce neuron in motor pathway. And here is our alar plate and rhombomer. Our cerebellum will develop from this part. As we can see in embryo at E9, this is the alar plate of the rhombomer, and here is the isthmus, and here we have a mesencephalon. At this part, I want you please pay attention that our trigeminal nuclei, they develop from our alar plate all the way from the mesencephalon, as you can see in here, rhombencephalon, to the upper, in fact, the cervical spinal cord. One of the important points is I couldn't find any information what happened to our continuation of the our trigeminal nucleus in the alar plate of the rhombomer 1, and this is the, I think, missing piece of the piece of the R puzzle during the R neural tube development in early stage of the embryonic development. So we have, in fact, another way to see that how our trigeminal system associate to the our cerebellar primordium. At this stage of the development, we have two type of the neurons. This is the Purkinje cell GABAergic developed from the ventricular zone. And here is this one is the cerebellar nuclei neuron that developed from the rhombic lip, the GABAergic one, and the glutamatergic one, and coming in here. But there is evidence and there are data showing that some neuron from the mesencephalon, they migrate and end into the cerebellar nuclei neuron in here. And I think and I believe this is the extension of the our mesencephalic trigeminal nucleus at this stage to the our cerebellar nuclei neuron in here. So whatever it is that these two sets of the cells, either cerebellar nuclei or the Purkinje cell, they are the only neuron that we have in here, but the individual neuron, they are not functional. In fact, they have to make an axon and connection and make a connectivity in here, either inside as intrinsic connectivity or the type with the outside of the cerebellum as the extrinsic connectivity. To understand this, in fact, process, let's see history about the what is the earliest neuron and axon development. In fact, this is the interesting issue for our researcher because they provide the foundation of the whole circuit formation or connectivity in the nervous system. In about the late 19th century through the 1940, it was believed that the basal plate developed earlier than the alar plate, and the MLF was the first track to appear. However, this one changed recently, and this one published in the PNAS and then in the Journal of Neuroscience in 1993, that the first track developed from the mesencephalic nucleus of the trigeminal nerve, a tract in alar plate, not the basal plate. That's the big change, in fact. Yeah. So, in fact, the mesencephalic nucleus of the trigeminal nerve is close to our isthmus. Isthmus means, in fact, in the isthmic organizer place. And then this organizer linked by multiple signaling mechanisms that control the development of the rostral cerebellum and caudal mesencephalon. And in addition of this relationship that they, in fact, provided this information, it was a long time ago, 1960, cerebellum without vermis. In fact, this is the midline cerebellar defect. Both vermis and the medial cerebellar nuclei, they are not forming. And this defect accompanied with absence of the mesencephalic nucleus of the trigeminal nerve. As we can see, all of this evidence indicate that the cerebellar primordium and trigeminal system develop in close proximity. But how and what is this relationship between the cerebellar primordium and trigeminal nerve in early development? This is the one that we are going to discuss in few slides later. So to understand this relationship, let's go what is the currently we know about this early connectivity. This is the cartoon simplified and coming from this level of the R section and rhombic lip in here. And this is the source of the R cerebellar nuclei neuron that migrate to the nuclear transitory zone in here. And here is the neuroepithelium that they form the Purkinje cell. They located in Purkinje cell plate in here. Conventionally, it was believed that the vestibular afferent are the first to invade the cerebellar primordium and terminate on the Purkinje cell somata. This is the one that, in fact, we believed. And however, I put the cross line in here, and we made a hypothesis that the trigeminal ganglion pioneer axon enter the cerebellar primordium before the Purkinje cell are born, where they seem target the cerebellar nuclei neuron. Our Purkinje cell born around the E10 to E13, but our cerebellar nuclei, they born at E9 to E12. So to understand more, I dissect the cerebellar nuclei in this box in here. And we know this part. This is the neuroepithelium making the GABAergic cerebellar nuclei neuron. This is the rhombic loop, glutamatergic one. But the challenge is this part, the involvement of the trigeminal system. How are trigeminal system involved in early connectivity in the cerebellar primordium? Well, first of all, we have cells probably coming from the mesencephalon in here. This is the cells coming from mesencephalon, crusty, smooth, and ending to the nuclear transfer zone and intermingle with the cerebellar nuclei neuron coming from the rhombic loop in here. And apparently, this group of the cells probably is suggesting that may they are coming from the mesencephalic trigeminal nucleus, or maybe modified version of this nucleus in the cerebellar nuclei neuron. And we know there is lots of this suggestion or information that probably they are, in fact, forming from the neural crest cell. In the other hand, we have a ganglion, trigeminal ganglion, with the two part, this one, the placode part, and this is the purple one coming from the R, in fact, the neural crest, and they project to the cerebellar primordium. And apparently this one, as we can see in here, they form from the, in fact, part of them develop from the neural crest, means the neural crest are involved with somehow, there is some role for the neural crest in here. So, but whatever it is that I am just going to focus in early development for the connectivity, the ganglion project to the cerebellar primordium, probably cerebellar nuclei, means I'm just going to talk about this part, not this part that is we are working on. So to, in fact, understand our cranial nerve organization in here, we use the neurofilament associated antigen 3810 antibody that express in our trigeminal ganglion in here, our cell body neuron in here, and the axon in here. As we can see in our cranial nerve, the peripheral projection and the central projection in here. This is the our cerebellar primordium. This one is the our mesencephalon, and this one is the our trochlear nerve in this part in isthmus in here. So my focus is the our trigeminal ganglion. This is the trigeminal ganglion in the adult we can see in here. And this one, during the early development, the central projection, they pass through the our cerebellar primordium, probably reach to the our mesencephalon, or from the mesencephalic trigeminal nucleus, the axon pass through the cerebellar primordium and reaching to the ganglion to get the target tissue in here. The another part that I want to be focused today is the vestibular system. This is the our vestibular ganglion, and this is the otic vesicle in here, and our ganglion, vestibular ganglion, formed from this, in fact, the epithelium of this vesicle. So we know that in adult, the close relationship between the vestibular system and the cerebellum, even we have a part as a vestibulocerebellum. This is well established in adult. However, it seems that in early development, this is not the vestibular system that organizing our connectivity or the, in fact, the neurogenesis at this stage. This is the trigeminal system plays significant role to establish for the early connectivity in the cerebellar development. So to go to the next stage is the which pioneer axon are the first to invade the cerebellum. We have to compare the order of the cranial nerve development. So we have a tools in here, and this is the E11, and we can see trigeminal ganglion well developed, and this is the central projection in here toward the cerebellar primordium, and this is the descending one, and this one is the autocyst, and this one is the our vestibular ganglion. We can see the same in our section as well. So let's just please focus in this part, the vestibular ganglion and trigeminal ganglion. Let's go to early stage in here at E9. Well, we have our trigeminal ganglion that emerging, and our central projection from the trigeminal ganglion passed to the cerebellar primordium and probably to the mesencephalon, and maybe from the mesencephalic trigeminal nucleus, the axon passed from the isthmus and ending to the R trigeminal ganglion. But at this stage, close to our otic vesicle in here, there is no vestibular ganglion. Let's see more detail in here. This is an arrow we can see in here. This is the otic vesicle. This is the geniculate ganglion or for the vestigial nerve. And we don't have, in fact, the vestibular ganglion at E9. The time that the trigeminal ganglion present, even the ophthalmic branch, they are extending, and the central, in fact, the projection, they are passing through the cerebellar primordium. And even we go to the, with the confocal imaging, we can see most the rostromedial part. This is the extension of the fiber from the trigeminal ganglion to the cerebellar primordium. So that's the interesting. So, but the question was, do we have the direct projection from the trigeminal ganglion to the cerebellar primordium? This is the part we use the dye track tracing approach. This is the dye in the ganglion in late E10 or early 11. And we can see that this is the our fourth ventricle, this is our cerebellar primordium in here, this is the ventricular and subventricular zone, and this is the extension of the fiber that label with the diode that we have higher magnification in here. This is the one that you can see they are coming and then ending to our cerebellar primordium that we will see where the target the some cerebellar nuclei neuron here. So in addition, I want you, in fact, please attention that we have this numerous this collateral branch in this experiment as well that we will see in our immunohistochemistry as well. This is the NAA staining that we can see. This is collateral branch to this area that this one we will see is location of the our Purkinje cell. But are they are not going to answer correctly if this is coming from the trigeminal ganglion or this is from the mesencephalic trigeminal nucleus axon backward projection to the ganglion to get the outside. Why this is happen? Because our mesencephalic trigeminal nucleus is the unique in whole nervous system. In fact, first order of the neuron, they should be in our ganglion, but they are located in central nervous system. Yeah, that's the one of the question mark. Probably they are coming from the neural crest as well. So whatever it is that when we have this experiment, we can see our trigeminal, the mesencephalic trigeminal nucleus axon, they will back to the ganglion and go outside. And then some of them, they are projecting to the cerebellar primordium. And when we have our experiment with the dye, we probably, in fact, labeling both ascending primary projection from the trigeminal ganglion or descending projection from the mesencephalic trigeminal nucleus, or both of them. So to understand and confirm our observation and distinguish between these two sources of the early afferent to the cerebellar primordium, we use the neurogenin-1 null mouse. Neurogenin-1 is important for trigeminal ganglion neuron development. In fact, both placode and crest origin of this ganglion, both are depend on neurogenin 1 function. So if we have a neurogenin 1 null mass, and if we don't have a trigeminal ganglion, so based of our expectation, we should not see any projection to the our cerebellar primordium. Otherwise, we failed our hypothesis. Let's see, and this is that E9 that clearly we can see we do not have the trigeminal ganglion. We do not have any projection to cerebellar primordium. That's the beautiful one. When we compare in here, this is the, in fact, the littermate, wild type littermate in here. This is the otic vesicle. We don't have a vestibular ganglion as we proved it. And this one is the trigeminal ganglion projection to the cerebellar primordium. And this is the mesencephalic trigeminal nucleus. And then axon probably pass and back to the ganglion. When we do not have a ganglion, not only we do not have a projection any to the cerebellar primordium from the ganglion, in fact, the axon from the mesencephalic trigeminal nucleus, they cannot reach the ganglion to go to the outside. Means they probably use this, in fact, as a substrate to reach to the ganglion in here. But whatever it is, that this one showing that our earliest afferent probably coming from the trigeminal ganglion. And to test further this hypothesis, we use the embryonic culture. Well, this is the E9, and we put in vitro for 4 days, and they develop some landmark you can see in here. For example, or the nerve they develop. For example, this is the oculomotor one that we usually don't have in the E9. Trochlear nerve between the cerebellar primordium and mesencephalon at level of the isthmus we have in here. Our trigeminal ganglion, in fact, the peripheral branch they developed, even our vestibular ganglion that we do not have E9, they already apparently committed, and then we have a, in fact, a development in here. So whatever it is, take home message of this one is that our trigeminal ganglion directly project to our cerebellar primordium. So the conclusion for this part is that the earliest afferent target the cerebellar nucleo primordium originate from our trigeminal ganglion. The next is, what is the target neuron in the cerebellar primordium? Well, at this stage, we have a Purkinje cell, as you can see in here, the marker FACSP2 and PTF1N. 1A, and then cerebellar nuclei in here, the marker is the TBR1A and LMX1A. So this is the only cells we have, neuron we have in here. So conventionally, it is surmised that the Purkinje cell are targets the earliest afferent to the cerebellar, to the cerebellum. So let's see if this is correct. Well, if we take a section at this is at E11 at a stage of E11 embryonic day 11, we can see that in lateral section we can include our vestibular ganglion and trigeminal ganglion, and this is the LMX1A that in fact staining our cerebellar nuclei. When put together, we can see that our fiber at this stage at E11, we have a multiple source in here. They are ending to our nuclear transfer zone or in the cerebellar nuclei neuron in here. So if this is correct, and if we go to earlier stage at the E9 that we have just the trigeminal afferent from the ganglion, trigeminal ganglion, as you can see in here, they are terminating to area of the immunoreactivity we have with the LMX1A, the cerebellar nuclei neuron. At E10 that I didn't show in here, at E11 we can see again, this is the LMX1A neuron that they targeted by the earliest afferent. And also at E12, we can see with TBR1, another marker for the cerebellar nuclei neuron, that they are ending in here. And surprisingly, we can see huge number of the collateral branch in here that ending in this area. And this is the area that will be our Purkinje cell plate, means location of the Purkinje cell in here. So I will show you in couple of slides later in detail, but in here that we have a earliest afferent, they come to the cerebellar nuclei and they are make a contact in here. This, the way that the talk between the end of the R earliest afferent to the cerebellar nuclei is such as the finger-like process, and means such as the gross cone that may the ending in here, not just the passing through this nucleus. However, the collateral one, that the fiber coming this area, we mark them with the PTF1A. This is the Purkinje cell plate, and this is the FACSP2. And in detail, we can see this group of the fiber, they are ending in the Purkinje cell neuron in here. So in conclusion for this part is the pioneer axon target to the cerebellar nuclei neuron at E9, and later collateral pioneer axon projection target the Purkinje cells. This is the graphic conclusion that I think this is just some of them we showed in here. This is the earliest afferent. They are coming to the cerebellar nuclei neuron, and this is the efferent project outside. And this is the collateral ending to the the cerebellar cortex neuron, in fact, means the Purkinje cell. So if we, in fact, remove a few cells from the cerebellar nuclei and then the Purkinje cell plate neuron in here and label them, for example, ABC in here, our story is that the early afferent, they come to the cerebellar nuclei neuron and then project the efferent to outside. The collateral one, they go to the our Purkinje cell and probably they see from the cerebellar nuclei to see the Purkinje cell, and A and B the same that we can see in here. From now on, this is the one that we have seen some, but from now on, this is suggesting, or this is probably the best way to interpret that probably that the axon of the Purkinje cell, they in fact follow this collateral branch and then ending to the cerebellar nuclei that they originally we had from the earliest afferent, and we can then they establish our foundation of the connectivity in here. And this connectivity, we can follow it even postnatal, that we have our Purkinje cell monolayer formation in here. You can see the earliest afferent, they are coming, and then they, for sure, as a scaffold, the other afferent, they will come and join, and then efferent going out, and collateral, in fact, they're going to the Purkinje cell, ending the Purkinje cell, and the axon coming the same from Purkinje cell, the same cerebellar nuclei neuron. And this establishment, in fact, followed by the interneuron migration or connectivity, means the rest of them, this is the principle of the R, in fact, the functional module in here, or functional unit in here, that are interneuron, they follow this module. For example, one of the interneuron we have, this is the Ganol cell, and this one is the territory of this Ganol cell. They leave this axon as a parallel fiber in here and migrate to the Ganol cell and bridge between the afferent to the Purkinje cell. And this is the, in fact, they just follow the already established our connectivity in early development. In fact, in the early development, our axon, in fact, navigate the target neurons, but when they establish this pathway, later on, we can see our interneuron, our interstic connectivity, they just follow this established, in fact, functional unit during the postnatal development. So I would like to, at the end, I would like to thanks to my student, lab member, and collaborator for this study, and also thanks to our funding agency to supporting my research. I will be happy to answer your question, and I think we have a discussion session. We will meet over there. Thank you, and I am done.